Today we are going to discuss about genetic disorders, and these are illnesses caused by one or more abnormalities in the genome. And as you all know, some are heritable and are passed down from parents. That's from parents' genes. Others are caused by new mutations or defects in DNA. In such, they will be only heritable if they're affecting germ cells. The same disease, just like cancer, may be caused by inherited condition in some, by new mutations in others, and non-genetic causes in still some others. So today we are going to concentrate on the ones which are caused by genetic disorders. And to start off, we need to know where the genes are. Starting from way left, we know that a human being is an organism. It has cells, and it's within the nucleus of cells that will contain chromosomes. And these chromosomes on certain areas are where genes are located. So each chromosome is one long DNA molecule, and genes are the functional regions of this DNA. And as you know, DNA is a double helix. And to go deeper into it, as you can see here, this is the cell, that is the nucleus, and that's where the chromosomes are. And if you look at this, I hope all of you are seeing me. Are you seeing this on the screen? So the chromosome has a long arm and a shorter arm, and they're joined at the center, the area which is called the centromere. These are the telomeres. The short side is always referred as P, and the long side is always referred to as Q. And within the chromosomes, there are proteins which stabilize the chromosome itself, the DNA, and these are called histones. And as you know, that DNA is a double helix, and from it is where we get the genetic material. So chromosomes, here is where DNA is packaged. And DNA is packaged into individual chromosome along with proteins. The proteins, we call them histones. So DNA plus associated chromosomal proteins are referred to as chromatin. Prokaryotes are single-celled organisms lacking nuclei, and they have a single circular chromosome. While eukaryotes are organisms with nuclei. They have species, and every species has a specific number of linear chromosomes. And from this diagram, you can see that DNA is a double helix, and there are two of them which are intertwined, and they contain various sugars, as you can see them there, C, G, T, A, and these are bonded together with hydrogen bonds. And the backbone of it is a sugar phosphate backbone. So there are approximately 3.2 billion base pairs in every cell that build up the human genome. The genes themselves form only 1.5% of the human genome. So a segment of DNA is called a gene. And this encodes for the construction plan for proteins. And that's why it's called the backbone of human beings. So DNA can be thought of as a blueprint for an organism. It's composed of small molecules called nucleotides. And there are 4 different nucleotides distinguished by the 4 base pairs, 4 bases. There's adenine, that's usually abbreviated as A, cytosine as C, guanine as G, and thymine as T. And hence, DNA is a polymer, is a large molecule consisting of similar units, nucleotides in this case. DNA is digital information. So a single strand of DNA can be thought of as a string composed of 4 letters, ACGT, that are all repetitive, depending on which molecule is going to be formed. And I'll stop for a minute to hear if you're getting me. Hashim? Okay, so is it clear? Do I progress? A gene, hence, is a unit of DNA that codes for a specific protein. Okay? And genes consist of two segments. One segment is known as an exon, and the other is referred to as an intron. So the exons contain the DNA code for the protein, while the introns, which are intervening portions, they are of unknown function. So for it, once it goes out of the nucleus for transcription, the introns must be cut out, okay? And exons put together, spliced together in the mRNA before it leaves the nucleus. The location of a gene on the chromosome is referred to as locus. At this moment, I would like to tell you about the genotype and the phenotype. Remember that there are two strands of DNA as you saw it earlier. Okay, if I may go back, back. So these are the two strands, and on either side you'll find similar genes on either strand. So these are referred to as alleles. And the alleles that are present represent the genotype of a person. The expression of the genotype leads to what's referred to as phenotype, or what is clinically apparent in the person. Genetic heterogeneity refers to the appearance of a common phenotype for several phenotypes. And hence mutation refers to alteration from the normal. These mutations, which are alterations, will later on are triggered by very many things. They could be physical, they could be viral, they could be chemical, and mutations are triggered by many things. And the mutations mainly affect two parts. They may affect genes that affect growth, which are referred to as proto-oncogenes. So proto-oncogenes are normal genes that then become oncogenes due to mutations. Proto-oncogenes code for proteins that help to regulate cell growth and differentiation. And hence proto-oncogenes are often involved in signal transduction and execution of mitogenic signals, usually through their protein products. In short, proto-oncogenes are the genes that affect cellular growth. Remember that then, if they affect cell growth and there's mutations, then there will be uncontrolled growth. Upon activation, a proto-oncogene or its products become a tumor-inducing agent, and these are what are referred to as oncogenes. So examples of proto-oncogenes are like the RAS, WNT, MYC, ERK, and TRK. So a lot of these, you're going to see them in books. The oncogenes, proto-oncogenes work through two mechanisms. They can be converted to cellular oncogenes either through quantitative or qualitative. The other mutations, which I'm not sure we are put here, are through tumor suppressor genes. Okay, and the key tumor suppressor gene, as you all know, is p53. So mutations affecting these two genes, either the proto-oncogenes or the tumor suppressor genes, are going to induce malignancies. The other thing you should know is genetic mutations can involve autosomes of the sex chromosomes, and we have talked about that mutations involving the sex chromosomes are going to be heritable. The effect depends on the function of the gene affected. From the word go, you should know that a mutation involving a gene coding for an enzyme appears as a recessive trait in the heterozygotes because one gene copy is present and enough enzyme is made to provide for sufficient metabolic function. And hence you need mutations in both for you to get any functional abnormality. A mutation involving a gene condition for structural protein appears as a dominant trait because one of the abnormal gene leads to formation of an abnormal protein. Many single gene mutations, there are many single gene mutations, and they occur in many forms. The common ones that you know about are deletions and insertions. Okay. So far, are you okay? If you have any questions, do you have any questions so far? Okay. So there are many forms of single gene mutations. There are those that cause deletions, and deletions is loss of a single base pair. So when there is loss, there is what's referred to as a frame shift, in that the reading frame is altered because of this single loss. You can also have an insertion where there is a gain of a single base pair, also resulting in a frame shift mutations. Other mutations are referred to as missense, where there is change in a single amino acid, nonsense, where there is a change in a stop codon. Remember that these triple bases code for what is referred to as stop codon. So if there is a change in this, then the mutation is referred to as nonsense. Then you can have a duplication, whereby there's an extra gene with more protein production. Remember, there is what is referred to as splice site. So on the gene, we say there are exons and introns. So if there's a problem in that splice area, you can have abnormalities, and this is referred to as splice site mutations. You can also have triple repeats, also referred to as increased tandem repeats. All these are different types of single gene mutations. So let's look at some of these. So the disorders can occur at the bigger chromosomal areas or as single gene mutations. So as you know, DNA is packaged into individual chromosomes along with the proteins which are referred to as histones that confer structural stability to the chromosome. You know that human beings have 23 pairs, 22 of which are autosomes, and the pair of XY are sex chromosome. Haploid, which is usually put as an N, describes a cell that contains a single set of chromosome, usually gametes in human beings. And diploid, which is two of that, is now the whole set that is found in the cell. So normal eukaryotic organism is composed of diploid cells, one set of chromosomes from each parent, and hence 46 chromosomes in human beings are referred to as 23N. And when we are talking about numerical abnormalities, okay, you can have euploidy, where there's a multiple of haploid number, and diploidy, where you have either one extra or one less. Okay, so chromosomal disorders can be numerical or can be structural. Okay, so numerical is when you have euploidy, where you have multiple, okay, or aneuploidy, where you have one less, like trisomy, or one more, like in conditions of monosomy. Structural is where you can have translocation, where there's a movement of a segment of a chromosome. Deletion, where you have a segment of a chromosome being removed. Inversion, so reverse of a segment within a chromosome. Duplication, where there's a repeat of a chromosomal segment. These are referred to as structural disorders. Chromosomal disorders affect 50% of first trimester miscarriages. So a lot of stillbirths you may reckon are due to chromosomal disorders. Some of the approximately 0.5% of live borns have conditions like Down syndrome, where there is trisomy 21, or Fragile X syndrome. So a lot of these first trimester wastages are due to chromosomal disorders. You can also have somatic cell abnormalities when there are cancers. So Down syndrome is one classic example of a chromosomal disorder. It's also referred to as trisomy 21. It's a result of an extra copy of chromosome 21, okay? And it hence abbreviated like that, 47, 21 plus, meaning at the 21 position chromosome, there is one extra, so plus. How does it present? It presents characteristic facial features. The children or the people who are suffering of Down syndrome have short stature, heart defects, and usually have some degree of mental retardation. They're often sexually underdeveloped and sterile. They are susceptible, okay? They're susceptible to respiratory diseases and usually have a short lifespan. Those at risk for Down syndrome are usually correlated with the age of the mother, okay? But can also be the result of a non-disjunction of the father chromosome at position 1. But commonly, age of the mother is high risk. So the higher the age or the higher the age of the mother at conception, the higher the risk of Down syndrome. This is a karyotype. A karyotype is, you can see that the chromosomes here have been put, and then you look for the abnormality, and in this karyotype, the abnormality is in chromosome number 21, where you have one extra chromosome. So this is the actual karyotype for Down syndrome. Are you with me so far? Are you aware about karyotypes? So you are able to picture out that this extra chromosome here is what makes up Down syndrome. That's correct? Okay, so that's an example of the commonest chromosomal disorder, which is referred to as Down syndrome. Let's look at a few other syndromes. We have trisomy 13, referred to as Pathology syndrome. We have trisomy 18, referred to as Edwards syndrome. Turner syndrome is XO. Then there is Creel-Duchamp, deletion of part of chromosome 5, Klinefelter's 47XXY, Prader-Willi syndrome, deletion of chromosome 15. In this, where the patients have hyperphagia, mild to moderate mental retardation, short stature, small external genitalia, and hypotonia. So all these are examples of chromosomal disorders. Some with euploidy and diploidy, and others have the structural abnormalities. The other thing that we need to know about are patterns of inheritance. These are often seen in Mendelian disorders where whole chromosomes are affected. And these can be divided as autosomal recessive and autosomal dominant, where the autosomes are involved. Or you can have those that are affecting sex chromosomes, and the main ones are the X-linked. So you can have X-linked recessive and X-linked dominant. Let's look at autosomal recessive. This pattern is typically seen with a mutation in a gene encoding for an enzyme, resulting in loss of function. So heterozygotes generally have enough gene product to get by, while homozygotes have very little of this, and hence the amount of product is equivalent to the severity of disease. So severity varies depending on the amount of product involved. The standard recurrence risk in this is 25%, meaning in every pregnancy, there's a risk of 1 out of 4 of the products getting disease. Typical example of autosomal recessive is sickle cell anemia. This is a common autosomal recessive condition. In this, you have hemoglobin being affected, and you know that hemoglobin is comprised of 4 globin chains that bind iron. 2 of the chains are alpha and 2 are beta. So a point mutation in the beta globin chain leads to abnormal globin that causes red cells to change shape, so they sickle, and a low oxygen concentration. The mutation is a point mutation whereby there's a substitution of valine for glutamic acid at position 6. of the beta globin chain, leading to abnormal conformation of this protein. And in this, as you know, both the female and the males have to have a mutations for you to get this condition. If it's only one part of that allele which is affected, then they get what is referred to as a trait. Are you with me up to that? Do we proceed or do you want to take a break? So that's an autosomal recessive. An autosomal dominant, this pattern typically occurs when a mutation involves a gene encoding for a structural protein. There can be dominant negative effect in which the product interferes with formation of a complex protein such that there's gain in function, in which the product leads to appearance of abnormal features. Typical or one of the classical example of an autosomal dominant is a condition referred to as marfan syndrome. So what happens in this, there's a gene referred to as fibrillin gene, okay? It encodes for a protein that's a component of microfibrils that form connective tissues, particularly in the aorta, in the eye, and skeletal system. So mutations in the fibrillin gene result in abnormalities in fibrous connective tissue where microfibrils form part of the structure. So affected persons are tall with long fingers, something referred to as arachnodactyly. They have loose joints. They also have ocular problems. And in vessels, they have this condition whereby there's a propensity of the aorta to rupture. So marfanoid features are these features that affected people present with. And as you can see, that protein referred to as microfibril is what is formed abnormally because of abnormalities in the fibrillin gene, mutations in fibrillin gene. So Marfan syndrome is an example of an autosomal dominant. You only need defects in one allele for that to present. So that's an example of an autosomal dominant. Then there are many, so you need to go and read about autosomal recessive and autosomal dominant conditions. There are many, and they're there in the books. You can read about those conditions. How they arise is what I've just explained to you. What about those that affect X chromosomes, the X chromosome? So in this pattern, females are typical carriers. And you know that in females, it's XX. Because of X inactivation, there leaves some functional genes available to produce product. So the females display only traits, they don't get disease. Depending upon how much product is needed to be healthy, some females may be mildly affected. Classical example of an X-linked recessive is hemophilia A. Hemophilia is also called bleeding. It's a bleeding disorder. An example of X-linked recessive condition is hemophilia A. It's a blood clotting disorder. In this condition, there is mutation in the gene coding for production of blood clotting factor 8. Okay. This disease illustrates the fact that the amount of product made can vary somewhat because in patients with hemophilia, there are those who have severe disease and there are those who have mild disease. So depending on the amount of factor 8 produced, then there is variable expression from mild to severe depending on the amount of product formed. All of them have this mutation, but the severity depends on the activity or the amount of factor 8 produced. When you're talking about X-linked dominant, you usually, theoretically, both males and females will be affected. But it seems that a double dose of the abnormal gene product may be lethal in utero, so virtually no males are seen with this disease. So very few X-linked dominant conditions are seen. The other things that we are going to see in genetic disorders are penetrance, meaning there are those people who get mutations but don't show and this is due to penetrance. Reduced penetrance means that the gene is present and can be transmitted to offspring, but does not produce the phenotype in the parent, leading to what is referred to as skip generation in a pedigree. So you may have people who have the mutations, but because of reduced penetrance, the offspring don't manifest disease. So variable expression is where the disease is present, okay? But the severity of gene expression varies from mild to severe. Remember that most disorders have multifactorial inheritance, meaning there's no one specific gene that's involved. Instead, multiple genes, in conjunction with environmental factors, interact to produce a certain phenotype. The recurrence risk for this multifactorial inheritance is above the average of 3%, but the amount is difficult to state precisely. For example, diabetes insipidus, diabetes mellitus, the non-insulin dependent diabetes mellitus, where family history, dietary influence, smoking, and obesity, among others, lead to development of disease. So DM has multifactorial inheritance. You could have a genetic disease, but interaction with that, smoking and obesity will interact to show, to bring up the phenotype of diabetes mellitus. Some of the molecular events that led to discovery of genetic diseases are one, the main one here is where Watson and Crick found out the structure of DNA, which is the double helix. Then there's this person called Paled who showed that there are certain, the site of enzyme manufacturing the cytoplasm in the RNA organelles, which is the factory for proteins referred to as ribosomes. And many, many others up to recombinant DNA, where gene cloning, others I'll leave you to read about. Okay? But the main clinical applications of this is in discovery of many genetic diseases. One in malignancies, for example, Philadelphia chromosome. This chromosome is whereby there's translocation of chromosome 9 to chromosome number 22 in chronic myeloid leukemia. And in this, the result is in chronic myeloid leukemia, you can test for these abnormalities. Also, in identification of diseases, for example, TB, we are now able to use probe that can be used to detect genetic information of a pathogen in a sample. For example, now you get sputum and you want to see if there's TB in it, you can use probes, okay, like GeneXpert and other PCR-based tests. We also have neoplastic conditions like leukemia. We can look for surface markers, and we can also use this DNA in identity testing, especially in paternal disputes, in organ transplants, whereby you want to determine whether a particular body fluid came from a particular individual. A probe is a short, relatively well-characterized nucleic acid segment used to search an unknown sample for the presence of complementary sequences through the hybridization process. This is called nucleic acid probe. So this probe has been used in many, many areas. For example, in immunohistochemistry, FISH, SISH, PCR, genetic mapping, DNA typing, all these are now in the pathology realm. Okay, and as you know, there's a human genome project which has identified all the genes and their activities. Okay, and now the key area that is now ongoing is use of embryonic tissues as spare parts in treatment of certain ailments, which ethically has not been approved. Okay. And there are many other gene therapy solutions. For example, cloning, which is more developed in plants and animals more than in human beings. So there's reproductive cloning, there's DNA cloning, and also there is therapeutic cloning. There's also what is referred to as, genetic silencing, where you want to activate an existing gene which could be abnormal. Gene splicing, okay, whereby DNA from an organism is cut and some gene inserted for recombinance, for example, in production of insulin and vaccines. And we also talked about forensic, where there could be paternity DNA testing, crime scene trace evidence, identifying parts, body parts in mass accidents, identifying remains or decomposed, and when you are following lineages where certain segments of DNA in certain groups of people is preserved. Okay, I would like to stop there.